The Institute for the Study of Peak States presents the 6th International Psycho-Immunology and Psychobiology Research Symposium. In this video, Treating Disease Using Psychobiology by Dr. Kirsten Lykkegaard.
Our next speaker is Dr. Kirsten Lykkegaard. She's our research director at the Institute. She's a doctorate in veterinary science, veterinary medicine, and has a PhD in pharmacology. For over 20 years, she has contributed to fundamental biomedical research within the fields of pain, obesity, and type 2 diabetes. As director of the research at our institute, the focus is on developing effective disease treatments using psycho-immunology and psychobiology. She is also investigating the causes of diseases of unknown etiology, such as Parkinson's disease and autism. Today, Kirsten is going to talk about treating diseases using psychobiology. Over to you, Kirsten.
Thank you. So I realized that we actually we haven't given a presentation sort of capturing our whole model theory and application in one go. And in the past year, we had such great opportunity actually writing all this together and publishing it in a 3-part paper. So what I'm going to do in this presentation is sort of tie model theory and application into one overview. So if you just think about what we do as a whole brand new way of understanding and treating diseases, it's based on intracellular biology, and it actually gives us the means to treat diseases and symptoms that previously have been considered incurable. The results that we get are fast, permanent symptom elimination, and we do that in hours instead of, if you think about normal medical, especially chronic diseases, instead of it take days, weeks, months, or even years, if ever. Treatment is drug-free, and our model says that it can be applied to any disease. We're so far applying it, as you saw with the previous presentation, to a lot of diseases, and we will keep doing that. And it also gives the solution to finding problems, to finding the cause of diseases of unknown etiology. So a lot of diseases, there are theories, but still no solution to them. By using this model, there is a different way of actually gaining insight into what causes disease. So far, we have, as Shane also showed previously, we have these diseases that actually have, some of them are still maybe in the clinic, but they all have been verified. So we have hearing voices, Asperger's syndrome. We have treatment for symptoms after concussion, Lyme's disease, vertigo and dizziness, and the dispersed focused part of the attention deficit disorder. And in pipeline, we have Parkinson's disease, OCD, autism, chronic fatigue, tinnitus, and diabetes type 1. And so all of this has been possible because we have this unifying model. It takes all the observations that we have done in the past and kind of tie it together in a cohesive understanding. It's an understanding of, and it's been published in the first part of this 3-part series. It's the primary cell model, and it says that our experience, our everyday experience, is a mixture of that we have in our bodies, mixed with the sensations, feelings, and perceptions from inside the primary cell. The primary cell is a cell of, it's a totipotent stem cell that forms at the 4th cell division, and it's where we experience ourselves. And if you think about that, anything inside the cell that either is right or not right gets sort of mixed in with the experience of your body. That means if you have something in here that isn't right, it does feel like something in your body isn't right. And the other way around, if it's very healthy, you might have really great peak states. So in a sense, you can say that your body has a superimposition of the primary cell on top of it. And with this model, if that is true, we can draw a lot of implications, and we can make testable treatments and tests to see if it's actually true. So what we've done is exactly that. And building on the model, we have built this subcellular psychobiology theory. And as I said, with the superimposition of the two, it means that physical biological problems in the primary cell are experienced as physical and psychological problems, as sensation, feelings, and perceptions inside, on, or outside our bodies. This allows us actually to draw out unique symptom patterns, and they can be experienced kinesthetically, or some people even have visuals of what's going on. We call those subcellular markers, and they can be used diagnostically to target the treatment. You know when you're done, when the marker is gone. And with that, we actually see symptoms eliminate, again showing that the model, showing more validity to the model with that. We also observed that all biological problems in the primary cell are caused by trauma. And that can be directly or it can be indirectly. I'll show that in a minute. All traumas are rooted in epigenetically inhibited gene expression, and they can be fixed by trauma healing. That's also, again, a statement, and we have gone over it with all our work, and it seems to really fit. That's how they work together. So what is a trauma? So if you think again that things that structures and problems in the primary cell reflects into our being, you can see that traumas, they cause everyday psychological problems. That's the definition of a trauma. And simply said, it's a stuck emotion from the past, and you feel that it's from the present. So the example over here in this picture is, for example, an argument. So here you are arguing with somebody and you're pretty upset. You think this is about the argument, but in reality, what has happened is that in your primary cell, you have accessed what we call a trauma string. It's an inhibited or stuck mRNA string coming out of the nuclear membrane. So here's the gene. So the mRNA string sticks out in the cytoplasm. And if our awareness gets in, you can say, contact, if we put our awareness in here, this is where we get a flicker of a different situation. So actually this situation is building on emotions that come from a very early moment back in time. Here's a sperm. So this is just an example of how it builds up and how you can access different moments in that has the same emotions and feelings. So you can say this stuck mRNA string as a structure and trauma, they're the flip side of the same thing. Stuck emotion, stuck trauma string. It's caused by an epigenetically inhibition, but you can see here that we already have part of the string showing. Normal inhibition just doesn't make the gene have any transcription making this mRNA string. So something else inhibits this junction down here. We also know that effective trauma healing eliminates both the feelings and the images of what's going on. We still have a memory of standing in this situation, discussing things. Here we have a situation where we did fall and so on. But the trauma load has disappeared once this trauma string is released and the gene is now again functioning. So that's our understanding of trauma and how it actually plays out in our awareness and accessing it in the primary cell. So if we think about that in the model here, diseases and symptoms can only exist in our bodies if there is a physical biological problem in the primary cell. So what can these problems be? I mean, everything, as I said, comes down to trauma. But at the level of the present moment, you can say that your symptoms comes from either two types. So there's traumas, they cause everyday psychological, emotional issues. There you have a direct cause to trauma, you can say, because the trauma feeling that you have if you're upset in the discussion that I showed previously is the feeling that's in that trauma string that needs to be healed. When you deal with intracellular pathogens and the problems that they can give us with symptoms, the symptom that they give us is not the symptom that's in the trauma string. That's why we call them indirect traumas. You need to heal something entirely different. And that's what we do in research, to figure out what causes us to hang on and have this problem. So both of them, trauma. The problems from pathogens show up in three ways, as so far we've seen. We can have direct physical damage from a pathogen. That could be a rip in the primary cell. It can be felt in the body as pain. We can have psychoactive toxins. We have definitely diseases that actually are due to this. So OCD is one. We also have other infections that have toxins that are actually psychoactive in this way. We also have indirect that we can get be influenced by psychoactive structures. I'll show examples in this presentation using the Asperger's process or Asperger's biology to show you that. And if we then think about the psychobiology and psycho-immunology, there are three ways, in sense, if you kind of group them, that you can eliminate symptoms. So there's our original psychobiology, call it psychobiology 1, or actually PB, psychobiology. So I'll be using PB for some of this. So PB1, it uses all kinds of trauma healing techniques. It's what Grant wrote up in the book of subcellular psychobiology. So it has everything from trauma to simple techniques to get rid of things on a one-by-one basis, usually. They can be very easy to use, or they can be very hard, especially the ones that have regression, music, and phrases that it's tedious to get through. Later today, I'll also present on psychobiology version 2, which is where we go in and actually, we could not have done this version unless we had already cracked psychobiology. So in this PB2 version, we actually we go in directly and address the traumas that then eliminates the interaction with the specific pathogen. That means you don't get rid of the pathogen, it's still there, but the way it's producing the symptoms in our bodies disappear. So it's actually very, very cool. Then we have psycho-immunology. That's very cool too, and we will use that for certain diseases because you just want to get rid of a pathogen. They're bad news to our organism. So with that, we can actually heal the traumas that make our primary cell no longer want to house or harbor these specific pathogens. I've written in here that they're pretty hard. I mean, sometimes they go pretty quickly, but sometimes it can be very tedious. So we're still working on how to make this easier. Okay, so now comes the third paper in this three-part series. And that's where we are showing applications. So I'm going to show what we have in this paper. We have 3 examples of diseases that currently there aren't a good cure for. One of them is dizziness. I should have put in, it's actually vertigo dizziness. It's hearing voices, also can be thought of as intrusive thoughts. When they're not ramped up to voices, they can just be very intrusive. And then Asperger's syndrome. And I just want to just say again, remember the psychological client, emotional issues, which is easy, usually trauma, versus actually psychiatric and medical disorders of unknown etiology, where the symptom isn't, the one that you can heal. So in this case, you can't just heal a trauma string on dizziness. Actually, you can, but it won't solve the core issue of this problem. Same thing with hearing voices and Asperger's syndrome. Their sensation in the body is indirect to trauma. So I'm going to show you one by one here. So the treatment of dizziness here. The subcellular markers, so what's going on in the primary cell that we overlay with our body. In this case, it is the actual sensation of spinning. It is the marker, because what happens is that your awareness gets caught in the cytoplasm in the area of a damaged mitochondria. When this mitochondria is damaged, usually it sucks in fluid and squirts it out, so it's sort of like a pumping mechanism. And when there is what we call a vortex, that function has been screwed up. And what we see in this is that inside this mitochondria, the DNA rings, we believe it is, they are actually not functioning. They're stuck. And they actually feel like they're stuck to the bottom. If you go down, if you say this is the top, this is the bottom, they're stuck down on the bottom. And they are stuck down there because there is a foreign, very coding onto the gene structure. And so it's totally frozen. That's the feeling that it has in there. Yeah. So I just want to add that when you have this symptom, you can be going around like this if you're in the periphery of the spinning, or you can be in the center. And with that, you actually feel like your surroundings are spinning, as if you're in the center of the tornado, the 0 point. So how do we treat it? In this presentation, I'm just showing the one by one, because a person can have anything from a few to many of these. So, and actually what I needed to say also here is that we get into this whirlpool in the cytoplasm by triggers in our lives. And some of those triggers could be like excessive drinking. It could be other things too. I'm not sure exactly which, but movements, some of the disorders of the vestibular system could in fact be caused by this. We have seen a or not just improvement, elimination of something like benign paroxysmal positional God, what's it called? Dizziness. So the one where if you move your head in certain ways, you elicit this spinning sensation. And they believe it's a otolith that has been going out in the lymphatic labyrinth. So in the lymph, in the labyrinth, and with this treatment, that vertigo also disappears. So what you can do on a one-by-one basis, you guide the client to the bottom, you guide them through the funnel here, and drop down into what feels like a room and down to the floor. And in there, you have them find what feels like a structure that's damaged on the floor. And then by just feeling the discomfort in that structure, it will dissolve, and boom, the mitochondria repairs and that vortex spinning sensation disappears. What we recommend is that you have the client look for other vortices so that they don't just go out your office and evoke another one. And that's where later, when I show the one that eliminates all vortices, is a really plus to this treatment tool set. That was vertigo. If we take Asperger's syndrome, and again, the focus here is to show that we have a subcellular marker. People with Asperger's, they feel cut off from the world kinesthetically and also emotionally, that they can't feel what's going on in other people. They can't read facials or body language. And the reason is this glass wall they're in behind. And this is an example of a pathogen-induced structure. So it feels like a hardened structure around you. It is a bacterial covering on a primary cell structure in which we actually overlay on our body. So of course there isn't a glass wall outside our body, but it comes from this overlay. And the treatment of it is to actually healing the originating event where this film comes on, and you have to heal it on your egg side and your sperm side. Alternatively, we have a way that you can heal it directly on the marker, so on that feeling on the glass wall. And with that treatment, or both treatments, the Asperger's symptoms disappear immediately. It's quite amazing to see that people all of a sudden can actually feel their surroundings that they weren't aware of, that they couldn't do, except for they were aware that they were disconnected from other people's emotion, but seeing that they can see how tall the sky is, for example, that's been a very awesome thing of this one. And the fact that they don't need to learn to interpret body language and these things is quite amazing. There are also other people can actually feel that they now can connect to the person, which is another cool thing. Next is the biology of hearing voices. And this is a widespread disorder in the population. Actually, I have a reference here that 94% of the general population have intrusive thoughts, according to this study. And what do I mean with intrusive thoughts? I mean some kind of, you think it's your own thoughts, but it's actually something that you can't really just say shut off, as in a wonder. If you're actually thinking, you can just say, oh, I'm wondering, and then you stop wondering. Or but with this, it's just, it keeps going. But the spectrum disorder ranges from background thoughts during meditation to thinking that, or feeling that you can hear something coming from someplace, from somebody, maybe what's called channeling, to intrusive thoughts that can be after birth, for example, that women experience a lot of like thinking that something will go wrong with their baby, or they even have thoughts that they want to harm their baby. It's thoughts that they don't want to have, but they can't stop it. That's in this category, the same biology. And then to hearing voices in schizophrenia. So the subcellular marker is a thought or a voice, and it's in a fixed location, usually outside your body, usually outside the head. And its voice has the same specific unchanged emotional tone, like worry, disgust, anger. And the reason that we have them outside is that we actually superimposing the endoplasmatic reticulum where these ribosomes are sitting, and these ribosomes contain, as shown in this one, contains that emotion of each of the, what we think is a voice. So we can eliminate those by using body association technique, and that's one of the very cool, small, or very quick trauma healings that you can do. And by doing the worst 1 to 3, most people really feel an improvement in life. The other way to do it is targeting the all the voices where they form in the ribosomes. That was what Shane was talking about, that we actually have now in our, it's not released, but it's in our testing, where we can just eliminate all the ribosomes that contain the voices. That's got another cool benefit, but the best one that we have so far in terms of how much you get out of it is the one that it uses to get rid of a treatment that uses a step in development where it actually can get rid of the pathogen, fungal pathogen, that is the reason for the ribosomes to actually start having this voice phenomenon. Yeah, and we have, Grant has this book that he wrote in 2017, and it has a lot of good stuff, and it has, for example, the body association technique as a free in the appendix. Yep, that's it. If anyone has any questions.